Next slide, please. Hi, and welcome to the next webinar in our Plantae webinar series. My name is Jason Padilla, and I'm your technical host for today's webinar. This webinar series is brought to you today by Plantae, the open online community for plant scientists powered by the American Society of Plant Biologists. I would like to give a special thank you to all our ASPB members who are joining today's webinar. Your ASPB dues help support make this webinar possible. For any of you who have not yet joined ASPB, you can join today using the discount codes %10 to receive a discount on your membership fee. ASPB members get early access to these webinars, and you can learn more about ASPB at aspb.org. Next slide, please. This webinar is recorded. We have so many other Plantae webinars that we've done in the past. So if you're looking for great plant science content, whether it be education-based, career-based, or scientific kind of theme topics, you can check out our Plantae YouTube channel to listen to and tune into many other exciting talks. Next slide, please. As you may have know, ASPB is the one who hosts the Plant Biology Conference that happens every year. Registration for this year's Plant Biology, which is taking place in Milwaukee, Wisconsin on July 26th to the 30th, is now open. It is a unique event that is positioned you to bring together members of the plant science community from all over the world, where you can acquire knowledge, network, and build your skills, and to learn more all about the exciting plant science that are happening. And it's open to all career levels, and you can learn more about it at plantbiology.aspb.org. Next slide, please. Before we get started, I'd like to go over a few housekeeping items to make sure that you get the most of your attendance today. If you're experiencing technical difficulties, please let me know about those using the Q&A box, or you can email me at jpadilla@aspb.org. There will be a Q&A, so if you have questions for our guest speakers, please add them in the Zoom Q&A section. Our moderators will be sharing them with our speakers during the Q&A session. And if you have trouble connecting or need to leave the webinar early, again, this webinar is recorded and will be made available on our YouTube channel and will be posted also on the Plantae webinar blog. And so you could kind of catch it later. And if you have friends or colleagues that would benefit from hearing this talk and can't be here today, please definitely share it and pass the recording to your network. All right, so without further ado, I am now going to turn it over to our Plantae Fellows who will be facilitating today's session. And so with that, take it away, Plantae Fellows.
Hello, everyone. My name is May. I am one of the Plantae Fellows that organized this webinar for particularly for undergraduates who are interested in switching from being always in their dorm rooms to experiencing science in laboratory settings. So this webinar will give our audience an idea of how and where to start thinking about exploring science during their career, and our panelists can share their personal experiences on how they got to where they are right now, which probably happened as early as during their undergraduate stints in their universities. And I know personally that science can be very intimidating, and our goal with the seminar is to make sure that approaching science as an undergraduate doesn't feel too difficult and feels more accessible to everyone. We have 4 amazing and diverse set of speakers, and I'm excited for everyone to hear what they have to share about this topic. We have Dr. Kevin Cox from WashU, Dr. Jason Rauscher from Corteva, Dr. Jess Gersony from Smith College, and Dr. Neha Bhatia from University of Cambridge. And we'll have each speaker present their presentations, and we have a Q&A afterwards. So if you have questions, feel free to put it in the Q&A box, and our speakers can pop in and answer those questions, or we can answer it live later too, depending on time. And lastly, this webinar was made possible through ASPB. And thank you, Jason, for helping us pull this off. And I would like to also acknowledge the 4 amazing fellows that's here, including myself. And I also want to say thank you to Malini, Gaurav, and Stephanie for being able to get this webinar together and for it to be as successful as it would be today. So now let's get this webinar rolling.
Hi, everyone. I'm Malini, and I'll be one of your moderators for this exciting session. Thank you all for joining us again. And without further ado, let me introduce our first speaker of the day, Dr. Kevin Cox. He's an assistant professor of biology at WashU, an assistant member at the Donald Danforth Plant Science Center. Dr. Cox's work explores how genes are spatially organized in plant tissues and how the spatial setup enables plant cells to communicate, especially under stress. Dr. Kevin, thank you so much for being here today. And please feel free to go ahead and share your slides. The screen is yours.
Awesome. I'll go ahead and share my slides. And let me go and start the presentation. Okay, everything looks good? Thumbs up. Nice, awesome. Okay, so thanks to the Plantae and Plantae fellows for inviting me out to give a short presentation. So I'm just gonna basically talk about like just like a one-click slide about like what my lab does and then go into like how I got into science. So overall, my lab, what we do is we try to focus on like how our genes are spatially organized and how they're taking information perceived from, let's say, any abiotic or biotic stress and regulating them across like plant cells and organism, or like an entire organism. And so what my lab basically does is we use like a variety of spatial and single cell biology approaches to look at specific biological questions. So one is which is like looking at plant pathogen interactions, and other is through duckweed biology. Okay, so how did I get into science? So I did my undergrad at the University of Missouri in St. Louis. So I'm a St. Louis native, so I was born and raised in St. Louis. Did my bachelor's in biology with a minor in chemistry. And when I had attended my undergrad, my initial career goal was to become a pediatrician. I had a passion like throughout the course of my life that I always liked helping people, and I always loved interacting with kids. And so I figured like the best way to do that was be a pediatrician, because that seemed to make sense to me at the time. During my sophomore year, I took a microbiology course, and that course alone changed my life because it brought me to like a new perspective, like what microbes they can do for us, how much they can benefit us, how much they can harm us, all these cool mechanisms that they can do. And so at that point, I knew I wanted to go into research and just have this like career shift from being like a medical doctor to somebody that like goes into research and study microbes for a living. So I had this new desire to go into microbial research. So I'm a plant scientist, so you might be asking like, okay, well, how did I get into plant science? So at the end of my sophomore year, I had a small dilemma. And what that means is that like while I was interested into going to research, I was working at this dollar store, working as a sales associate. Basically, it just paid the bills. Wasn't the greatest job in the world, but that's besides the point. And so I had to take a little bit of a risk because I wanted to see if I really wanted to actually go into research and actually do it for a living. And in order for me to do that, I had to kind of step out of my comfort zone and look into another lab that allowed me to do some undergraduate research. There was also a second risk to this. I had a kid at the time, or I was getting ready to have my first kid. And so me going into this undergrad research path would result in a pay cut. But thankfully, I had like a very supportive partner who's not my wife, who kind of encouraged me to go into research. So I started looking for undergrad part-time jobs, and I came across this job ad from the Donald Danforth Plant Science Center where I'm at today. At the time, I had no desire to go into plant science. I didn't really care for plants up until this point. But I just saw the ad up there, and I just figured like, why not? It's a way I can get experience. And I got the position, and that role like completely changed my life as far as like where I wanted to go into my career. So at the Danforth Center, I got really exposed to plant science. I got really fascinated by all the things that plants they can do for us, how much they benefit us, how they can defend themselves against microbes and stresses. And so when I got into plant science, I worked in a lab in Dr. Todd Mockler's, where like I worked on the Brachy transformation pipeline. And that I got experience. I interact with like grad students and postdocs, other lab members, and I was able to get their advice as far as like, you know, next career stages as far as like, grad school or industry. And it ultimately like resulted like in a paper that got published on. And so when I did my, so I was a part-time lab assistant from the end of my junior year all the way up until I went to grad school. So when I graduated from my undergrad and then like went off to grad school shortly afterwards. But I guess the cool thing about me having this part-time lab assistant job at the Danforth Center was it kind of really fueled me that, hey, I really like doing research and I really want to go into it. And it led me to the point to where when I started my senior year at UMSL, I had joined a professor's lab for research credit my senior year. So this was like more focused on bacterial pathogenesis, so very heavy microbial work. It allowed me to present my first poster at an undergrad research symposium, and it allowed me to get connected to like, again, more people and get their advice as far as like what grad school is like and what research is like. So a couple of quick lessons and advice I will kind of share with you guys. So one, I say to keep your options open. When I walked into undergrad, I wanted to become a pediatrician, but because I kept my options open, it kind of let myself steer myself in a direction to where, I desired to kind of go into plant science. Even though I didn't really care for plants initially, allow me to keep those options open and allow me to absorb different research opportunities, allow me to find a career in plant science. It's okay to be afraid to take risks, but don't let that stop you from taking some anyways. So I took a risk going from a paying job at Dollar General to like a not as much paying job to research, even though like a couple months later, I actually ended up making more than I did at the dollar store. So it kind of worked out. And it's also like a little bit of risk, like going up to talk to a faculty member, but sometimes you have to kind of take those risks to kind of get yourself to that next step. And speaking of which, don't be afraid to talk to faculty about research interests. I know we tend to come off as like a little bit intimidating because we're busy all the time and like, and everything. But to my experience, most faculty, they really enjoy talking to undergrads and kind of getting to know undergrads and like, figuring out like what their research interests are. And then lastly, this part sounds cheesy, but follow your passion. So my passion was like always helping people and helping others. And that kind of led to like my career where I'm still helping people and others. It's just in a different context. And so all this to kind of wrap up and say that like, I really happy that I kind of took the leap out of my comfort zone and worked in a lab during my undergrad because it provided me that needed experience. And today I have 2 undergrads that work in my lab and they're all great. And it's kind of cool just kind of making it full circle to where I can now give back and kind of help them like direct their career progression. And so with that, I want to thank you guys for your attention. There's my contact information and I'll be happy to take any questions like afterwards. Thank you.
Thank you so much, Dr. Kevin. That was a great talk, and thank you for sharing your insights with us. Now we move on to our second speaker, Dr. Jason Rocher, who is Academic Relations Manager at Cortiva AgriScience. Dr. Rocher brings over 2 decades of experience in science, education, and global academic engagement. We're excited to hear from you, Dr. Jason. Please feel free to share your slides, and it's over to you.
Great, thank you so much. And thank you for the invitation to participate today. I am going to give a slightly different perspective, that of somebody whose career spans a little bit longer than the other panelists, I think, here today. So I'm going to try to cover 35 years in 7 minutes. So I may be talking a little bit fast, but let's see how this goes. So let me just give you a brief overview of my journey. So I'm originally from upstate New York. I went to college in a small liberal arts college in Northfield, Minnesota, Carleton College. Got a bachelor's in biology there. But while I was there, as Kevin was mentioning, the importance of connecting with professors and doing research, that's exactly what I did. And I ended up working at a prairie in Minnesota. And that research that I did there ended up resulting in a publication and a passion for research and science and continuing on the traditional academic path. And so I went to graduate school. And again, connecting to Kevin here, I went to Washington University in St. Louis to do my PhD. And so I spent some time there. And during my research, I had the opportunity to do field work in South America. So I spent a lot of time in Venezuela and Colombia, studying these weird plants that you see in the pictures there. Because I was doing the field work there, I ended up having to do lab work in Colombia as well. So I ended up at the International Center for Tropical Agriculture in Cali, Colombia. And so a lot of my graduate career ended up not in St. Louis, but in South America. And so from Cali, Colombia, I eventually made it back to St. Louis, where you do have to finish up and do the writing and get your publications. And so I got my publication out of that. Again, right, this is the traditional academic path here. You do your work, you publish it. The next step for me was a postdoc, right? So I went to Cornell University, did my postdoc there, working in genetics, plant genetics there. You do your publications, you get opportunities from that. I did a Fulbright and I ended up back in Cali, Colombia. So I spent a few years returning to Colombia, teaching there, and doing research. That then led to an opportunity as again, this traditional academic path to be an assistant professor at the University of Puerto Rico. And so I spent a few years there. And then you have to make a lot of difficult decisions during your journey. A lot of them, involve specifically your career and decisions along those lines. Others are family related, right? So I had to make some difficult decisions, between the careers of myself and my wife and what was best for my family. So we ended up moving back to in this case, Pennsylvania. There are other stops along the way here. I'm trying to simplify it, but I was a visiting professor at Swarthmore College for a while. Then we ended up moving eventually we made it to Des Moines, Iowa, where I live right now. And that ended up in an opportunity to leverage my experience in research and education in a very different way for a large company called Corteva Agriscience. And we can talk more about this in the Q&A if you want to, but this was an opportunity for me to work in a space called academic engagement, where I run programs for students at universities all over the world. And so I'm engaged, traveling, visiting universities, interacting with graduate students and undergraduates as well. So kind of leveraging my 30 years of experience in a very new and different way. But I still have that passion for ecology and evolution and plant genetics. And so I do that in a little bit different way. So kind of like I worked in the prairie back in Minnesota way back when I was an undergraduate. Today I'm managing a 25-acre prairie here in Des Moines, Iowa in my spare time. I'm also a board member of the Central Iowa Orchid Society. So getting that passion for tropical plants and evolution and diversity in a very different way. So that's kind of my unusual path, right? So you can see it kind of started off as a typical academic path, and then it kind of went in different directions. And if you had asked me as an undergraduate, I would have had no idea where I was going, right? If you asked me as a graduate student, I would have said, yeah, I'm going to go the traditional path. I'll be a professor. That's my career path. If you'd say, no, you're going to end up working for a large company, I would have told you that, that sounds crazy, right? So here are a few thoughts on my journey, and this is going to overlap with what Kevin is going to say here. So the first one was Kevin's last point there: follow your passion, right? Know yourself and discover those passions. And then sometimes you're able to apply those in your career, and sometimes you apply those in your life and kind of your hobbies, your activities, things like that. So there are a lot of different ways to apply your passions. Be prepared and open to opportunities, right? So you seek out those opportunities. Some of them are expected. Some of them come totally out of the blue. You have no idea where they came from. They're totally unexpected. But by being prepared, your education and mindset, you're ready to take advantage of those opportunities. And I use the Louis Pasteur quote here: "Chance favors the prepared mind." That applies to serendipity in research as well as your career, right? So by being prepared, you're ready to take advantage of these opportunities. And then be ready to navigate change. It's very rare these days that you're going to be on one path, and that's it, and you know where you're going to be in 30 years. I can tell you, you probably don't know where you're going to be in 30 years. And so you need to be ready for change. Those decisions can be difficult, but they can also be exciting. You have to balance work and life, and you have to balance family, careers, all these different things. It's scary, but it's also exciting. It's fun. It's a big adventure. So the last point I wanted to make here for you all, especially, as you're looking to the scientific community, I think it's really important for us in this day and age, and I'm not going to make any political commentaries here, but to realize how complex the scientific community is and how these parts engage with each other, right? So we have nonprofit organizations, and Kevin, right, talked about the Donald Danforth Center and all these different organizations, nonprofits, interacting with international organizations, right? If you like international agriculture, the CG's system, like CIAT, where I was working in Colombia, conservation organizations, societies and professional organizations like ASPB working together. And then you have the government organizations, right? NSF, USDA, in other countries as well, places like Brazil, you have Embrapa. Then you have the private sector. You have, not all of these are startups, but small companies, right? And then large companies like Corteva, right? Multi-billion-dollar companies like Corteva. These are all interacting together, and the center of it is academia, okay? This is the only way that this system works. Progress, scientific research is dependent on these interacting parts, and they all have to be strong, okay? And they're all exciting opportunities for careers for those of you who are looking towards the future. So I will pass that. That's all I have to say, and we'll pass it to the next speaker.
Thanks so much, Dr. Jason. That was a fantastic talk, and it's really inspiring to hear about your journey of more than 3 decades. And there's an announcement, dear audience. If you have any questions for either Dr. Kevin or Dr. Jason, please feel free to drop them in the QA box. Thank you. And now it's my pleasure to introduce Dr. Jess Gersney, who is an assistant professor at Smith College and the leader of the Place Lab, where plant science meets art and community engagement. Dr. Gersney's research focuses on how northeastern US trees respond to environmental stress, and she's also a poet and former professional tap dancer, truly blending science and arts in unique ways. Dr. Gersney, please feel free to share your slides, and the screen is yours.
Getting it up. Okay, how does that look? Thumbs up? Great. Okay, yeah, it's good. Okay, thank you, Malini, and everyone for inviting me to come here and for being here today. It's truly a pleasure, especially to go after Kevin and Jason. You guys are very inspirational in many different ways. Okay, so my name is Jess Gersney, and I am in my 3rd year of being an assistant professor at Smith College, which is in Massachusetts in the northeastern US. And I run the Place Lab, which stands for Plant Physiology, Art, and Community Engagement, which I'll talk a bit more about at the end. Okay, so who am I? How did I get here? Hi, I'm Jess. I was born in Vermont, in Rutland, Vermont, on the land of the Abenaki, Wabanaki, and the Mohican, and I moved around a lot in the Northeast growing up. I grew up middle and an upper middle class, and I'm white, and I'm queer, and I'm a settler. And I went to, this is already where my journey starts meandering. I went to a creative and performing arts high school where I was a dance major, and I had the most amazing science teacher ever named Dr. Moon, who really started getting me to think about, okay, I love dance, but maybe I also love science. And so during this time, I was taking a lot of science classes in high school, and I was also tap dancing all the time. I then went to Oberlin College my freshman year, where I was like, okay, I'm gonna study science. And I really had this idea in my head that science is what I should pursue like institutionally, and you could always do art outside of institutions. So that was part of the reason and the thinking I had there. So I was like, let's go to college instead of a dance conservatory and take science classes and do some tap dancing also. But then I realized very quickly after my freshman year that I would like to try and be a professional tap dancer, and you cannot do that in Oberlin, Ohio. It's very, very difficult. So then I applied to every school I heard of in New York City, got into Columbia University and transferred there. where I was an ecology major and also took a bunch of poetry courses. And I also had my dreams come true and joined a professional tap company. You can see me there in the background, tap dancing. But at the same time, I started my research journey. So at Columbia, in the ecology program, at the time, they required you to do an honors thesis, which meant that you did research this summer before your senior year that was funded by the institution. And so this was a very, very cushy entry into research. And I didn't do any research prior to that. And I was really quite nervous about doing it. So I didn't do anything until the summer before my senior year, where I got to go to the Arctic tundra. Oh, I'm not wearing the exact same shirt, but it's a very close shirt to the shirt that's in that photo of me in the tundra. And I'll talk a little bit more about that. So I got to go to the Tulik Field Station, which is in the tundra of northern Alaska. And so you flew into Fairbanks and then drove 10 hours north. And there, it was awesome. I got to go for 2 months the first time I went, and that's me with the research team in the top right, which was a collaboration between University of Idaho and Columbia University. And there are a lot of awesome animals that are not plants, but are still sick. And so we saw this cool bear. We had this little baby fox who lived under where we slept at night. And then you can see on the bottom left, that's kind of what the station looked like. It was a lot of temporary structures that we that we made into research labs. And it was amazing. And I totally fell in love with research through doing this. And so like others have mentioned, I think getting in the lab or the field as early as possible is a great way to figure out if you want to go down the research path. And I absolutely fell in love with it. I could not believe you got paid to do this kind of stuff. And so that really kind of changed my trajectory. And so there's some more photos of me doing fieldwork up there. I loved it so much that I begged and pleaded everyone I knew who worked at Tulik Field Station to hire me as a research technician for the next year after I graduated from school. And I was lucky enough that the great Dr. Shahid Naeem, who's on the bottom right in his bug net, hired me for a year to be a research technician. So I got to go back to the Arctic again. Okay, so I was a research technician for a year studying climate change plant biology in the Arctic tundra. And like I said, that's when it really became clear to me that I wanted to go into science. And so during my year as a technician after undergraduate, that's when I applied to PhD programs. And I did that through talking a lot with different professors I had and trying to get advice from them as to where they think would be a good fit for me to continue my scientific journey. So then I ended up getting my PhD at Harvard University in the Holbrook lab, studying plant physiology. That's our lab up in the top right on one of our beach days, which was awesome. I also, when I was in my PhD, I continued my pursuit of art in many different ways. I took a bunch of poetry workshops while I was at Harvard, because you can do that. And so that was awesome. I also was in another tap dance company. And then once I graduated from my PhD program, I did a 1-year postdoctoral research at the University of New Hampshire, and then ultimately got this job as an assistant professor at Smith College. And so here are some photos of the lab. You can see me on the left doing some COVID research. My 3rd chapter I had to do in the heart of the COVID lockdown. So you can see me there masked up. I wasn't by myself because someone did take the picture, but I'm pretty sure it was just the 2 of us like in the whole building. And then on the right, I got to do some field work where I got to go to Chile and study Dolana mollis in the Atacama Desert. And this is the amazing group that I got to work with for those 6 years. And then here are some photos of me doing my one photo, the one on the left, the bucket truck, is from my PhD because I ended up studying northeastern trees and how they respond to drought. And then we spent a lot of time in the woods measuring a lot of things, and it's so fun. Now I run the Place Lab. So here is the images of all the amazing people in the Place Lab, where we're have a few different projects going on studying how plants respond to climate change. And then, so I guess just in summary, thinking about paths to research and any advice or opportunities I know of that I feel like would be helpful. So full-time path to research after you graduate would be things like looking for research technician positions like I got, which was such a gift. And you can find these types of things on this thing called the Ecolog listserv that I think we'll put the link in the chat too, so you can have access to sign up for it. These are pretty competitive and hard to come by, but they do exist. So I think if you're interested in it, it's something to try and strive for. There are paid master's programs. This was big for me. I did not know this when I was in undergrad. I assumed all master's programs you had to pay for, but there are a lot of paid ones. And there's another link we'll share with you that is kind of an aggregation of different programs that have paid master's programs, which is a great way of getting some research experience if you didn't get to have it during your undergrad, which is very normal and totally okay. And you can go straight to PhD. If you're one of the people who are like, I am obsessed with this and I know I want to do this for a really long time, then applying to a PhD during your undergrad is not a wild thing to do at all. It's very reasonable. But most importantly, it's also totally okay to meander. I think a lot of us have talked a bit about meandering, and like I have a friend who took 10 years between her undergrad and her PhD at Columbia, and she's an amazing scientist. And so you can, I think the idea of following your passion and where that ends up is really, and following that and ending up where that ends up is great advice generally. There's also summer positions, and some of these are specifically for US citizens, but there's also other ones that you can find. So there's the NSF REU program that has summer programs. The National Ecological Observation Network has a lot of summer positions. That's something to look into. And then Ecolog also posts a lot of summer positions that are in specific labs that are funded by grants, but they can take all different citizenships. So that's a great opportunity. Okay, I'll stop there. Otherwise, I'll talk forever. Thank you so much for listening, and I look forward to your questions.
Thank you, Jess. That's a great talk. And now I'm delighted to welcome our final speaker of the day, Dr. Neha Bhatia. She is a research group leader at the Sainsbury Laboratory, University of Cambridge. Dr. Bhatia's research dives into the development of plant organ forms using cutting-edge imaging and genetic tools to understand how leaves and shoots take shape. Dr. Neha, please go ahead and share slides. Over to you.
Yeah. Can you see my slides? I'm not able to see the panel at the moment, so can someone unmute and confirm that they see my slides?
Yes, we can see your slides.
Great. Thank you very much. So,
I hate to interrupt you, Neha. It looks like I'm seeing it in speaker notes mode. I don't know if anybody else is seeing that.
is that better?
Yeah,
It
this
looks
one
like-
works.
Okay.
Okay.
Yeah.
Great. Yeah.
Thank you.
Thank you. Yeah, so I am a plant developmental biologist by training, and I understood this assignment slightly different. So, having heard all these amazing journeys of past speakers who just presented, I thought that I would use my science to explain what inspired me to do science. So before starting, I would just like to very briefly show my academic journey so far. So I come from India, where I did my bachelor's and master's both in plant sciences. And then I moved to Germany in EMBL Heidelberg to pursue my PhD. And then while I was deciding what I want to do for my postdoc, I had a chance to briefly do a bridging postdoc in the same lab where I did my PhD because my supervisor moved to Australia. So I followed along and continued working on some of the other projects that I had started during my PhD. And then I came back again to Germany to do my postdoc. And then very recently, I've started my own group at the Sainsbury Laboratory in UK. So I have always been interested in understanding plant development from the perspectives of organogenesis and growth control. And even if it's plants or animals, I think that there are three core fundamental principles that define development: organ positioning, acquisition of special cell types, and then growing to correct size and form, which is morphogenesis. And I've been very lucky throughout my academic journey to have touched upon all these topics during my research. So, but what sparked my interest in plant development was this beautiful image that I still very clearly remember seeing in a lecture on plant development by one of my professors while I was doing my master's in India. So when I saw this image, I knew it was a confocal micrograph, but at that time I didn't know how to produce any image using confocal microscopy. I knew these were fluorescently tagged genes, but how these fluorescently tagged genes have been generated or are generated. So basically, on the technical aspects, I had no experience. And my professor continued to explain that what I'm seeing in these micrographs are actually patterns of a transport protein called PIN1 that mediates guided transport of the plant hormone auxin. And this protein concentrates auxin at these high spots of expression. It's polarly localized, as you see it in the form of these green arcs. And these cells sort of coordinate how their PIN protein will be localized in which direction, so they can bring auxin from all cells to concentrate at this site. And this is where these flowers or leaves emerge. What we see is as these broad scale patterns. And if this mechanism doesn't happen, then we don't get any flowers. And if we apply auxin locally, then we do get these flowers. So the big question in that field at the moment was how these polarities are coordinated. And while sitting in that classroom, I had never thought that I would get a chance to actually explore this question by joining the lab of the scientist whose image I was looking at and getting motivated. So I went to EMBL Heidelberg. I was lucky enough to get an interview call for the PhD international program. I didn't get the PhD offer at that time, and I started off as an intern in his lab, learned techniques, and then eventually got a PhD and worked on this problem. And 3 years of that PhD work summarized in one slide, we could finally get to an answer of that, how these polarities are coordinated, where we discovered a mechanism by where localized auxin signaling itself instructs neighboring cells to orient their transport protein towards these cells with high auxin signaling. This looks very straightforward, but it was those 3 years of almost blood, sweat, and tears dealing with this mutant that wouldn't even want to grow, doesn't have root. And then I would start off with 500 mutants at that time, and then try to keep them alive and then do experiments. But I found it very rewarding because these years of my PhD taught me many different techniques, how to use confocal microscope, how to generate images, how to do time lapse, how to do cloning, and how to interpret and read data. This was my very first graph that I made in like sort of a box plot, and I was so happy. So everything was very rewarding to see. And then most rewarding was to reflect back on this aspect that I saw this beautiful image in my master's, and 11 years later, I got a chance to actually investigate and understand a bit more that problem. And so this work led to an increased understanding that it's auxin-mediated transcription that feeds back on the auxin transport protein to generate auxin hotspots at the specialized structure called shoot, where we see new organs generating. So every time I see these kind of beautiful spiral patterns in a garden, I do get reminded of, maybe it's that mechanism that has given rise to these forms. And what was even more interesting to see that these mechanisms are so common that they not only initiate leaves from the meristem, but they also give rise to these complex leaf forms that we sometimes see, for example, a carrot leaf, or in wild cress, what we call cadamine. So then I joined Professor Milton Cianti's group at the Max Planck Institute to understand how these complex leaf forms arise. And then we knew at that time that this same mechanism of feedback loop between the PIN protein and the hormone auxin operates at these leaf margins. In addition, there are two growth repressing genes that form an additional mechanism of repressing growth along these margins. So auxin would drive increased growth, and then there will be reduced growth at these bases and notches that would enable separation of these units into these distinct leaflets. And so I decided to understand how these two proteins act, and we decided to investigate the expression pattern of these proteins in leaves. And when I saw this image after the confocal scanning was finished, I was just so amazed by this beautiful pattern that came out. It was, I still remember, 9 p.m. in the night, and I saw Angela, another group leader in the department where I previously worked, in the lab, and I was so happy to see someone that I could share these beautiful patterns with me. So with all these experiences, what I'm trying to say is that if you're really interested in what you're doing, you will really find joy even in difficult times during your research journey. And this led to designing more experiments, understanding more of these patterns through designing more complex experiments, and then generating genetic strains with perturbed function. We learned that when this expression pattern was abolished, then we don't get these characteristic leaf forms. And then because these two were growth repressing genes, we wanted to understand the underlying growth patterns, what they could function. So we used time-lapse imaging and then more techniques called sulfate mapping and quantitative cell level growth analysis. As easy as it sees here or looks here, it took me a lot of time to learn. So my postdoc was actually almost like a new PhD, learning very new techniques from the scratch. And then through all these experiments, we discovered that there is a division of labor between these two genes in a manner how they repress growth. So one limits growth but not cell division, and the other limits growth as well as cell division, and they play very different roles in making the complex leaf form. And what I find very rewarding about pursuing fundamental research is that it helps us understand complexities of life in general. For example, I get very fascinated when I can see how a complex leaf would develop in comparison to our limbs that have both each repeated units that are separated to some degree, and both have used some sort of similar mechanisms like self-organization that drive growth, and then limiting growth at the protrusion regions, be it digits or leaflets, but the mechanism of limiting growth is very different. Decreased growth in one case and cell death in the other. So, and that enables and helps us even appreciate that plants being so sessile can actually do these wonderful things to create one leaf form. And so, yeah, what I learned that was pattern differential growth is at the heart of the leaf form. And then now in my new lab, I want to understand further or seek a deeper understanding of the principles behind cellular decisions underlying patterning and morphogenesis. And I'm investigating the role of another plant hormone called cytokinin in that. And yeah, I have a lot of people to thank who have facilitated my academic journey so far. My PhD supervisor, my postdoc supervisor, my professors who taught me these amazing lectures during my master's, funding from the Gatsby and the Sainsbury Laboratory for giving me an opportunity to pursue what I want. And if I were to give one advice to the audience who are looking forward to doing academic research, I think focus and resilience are the two things that if you have, it will help you drive and move forward. So thank you very much.
Thank you so much, Dr. Neha. That was a great talk. So that wraps up our speaker session for today. A big thank you to all our speakers for sharing your time and expertise with us. And now I would like to turn things over to Stephanie to guide us through the next part of the session. Stephanie, over to you.
Thank you, Malini. And again, another big thank you to all of our speakers. I think it was a really insightful conversation, just hearing the different perspectives from everybody. So now we're going to jump into the Q&A portion, and the idea here where I'll just ask the question and we'll kind of go in order. I'd like to hear from each one of the speakers and get your perspectives individually on these questions. And so we'll begin and jump off with Dr. Cox. We'll go in order from that. Dr. Cox, Dr. Rauscher, Dr. Gersony, and then Dr. Bhatia. We'll just go in that order from there. And to kind of kick things off, I guess just kind of jumping off of Dr. Cox, one of your points about how initially you kind of wanted to go into the path of being a pediatrician, right? And I think that's very common for many undergraduates, that we think that we want to go in one direction with our career and something completely pivots, a few years in. And maybe a student wouldn't even realize that they have interest or passions in doing research in the first place if they didn't try it. So from all of your perspectives as a mentor, professor, et cetera, what are your suggestions to get students to just try research, to get them motivated in the first place? What are suggestions you have to get them to try it out? Dr. Cox, we'll start with you.
Yeah, I think what helps right away, and I understand it can be a little intimidating, is just reaching out candidly to professors, or like if you have a class that you're teaching, or if you have a class that you're taking of a professor class and you like the topic, just go talk to them about like, hey, what are some research interests that you could potentially go into? And I think once you start with those conversations, then you can ask like, hey, do you have any opportunities for like either summer undergrad research or like research over the semester, or like just anything just to get you exposed. But I think it's important that if you have to get exposed in that area first in order to validate that you really will want to go into that area. That's what kind of helped me, I think, from just listening to the other panelists. That's what's helped them as well. It's by getting exposed early, they were actually able to validate that, hey, I want to actually do this for a living. But I guess the first step is that you actually talk to the faculty. And that is a little intimidating, but like I was kind of saying in my presentation, most of us we don't bite. We're just stressed out most of the time, but we don't bite.
So I'll add to that, as basically kind of based on my experience as a professor at the University of Puerto Rico. Don't worry about whether that professor is doing something that you think you're interested in, right? So, you may think you're going to become a veterinarian, and like you say, well, why would I go into a plant lab, right? Well, for one, just go to a lab that looks fun, right? That's part of it. Getting any research experience, no matter what it is valuable. So it does not have to align with what you think your end goals are. And often that's it. There are surprising ways that it helps you, right? So, when a medical school is, reviewing applicants and they see that you're broad and you have broad interests and you did research in plants, that gives a different perspective from other candidates. It's really valuable. So be, just look for, I would just look for places where you think the grad students are having fun, right? That would be my criteria. And you go in there and you talk to people and you see, hey, is there any way I can come and get some research experience?
Oh, this is great. I totally agree with everything that's been said. And the only thing I would add is, I have a lot of undergrads in my lab that move in and out over the years, and I pretty much assume they don't know anything. And I'm saying that because don't feel like you have to be an expert in the topic to like ask a professor to join their lab. Like I have had students who have taken multiple courses with me, and they come into the lab and I'm like, oh, do you remember? And I'm like, don't remember, don't worry about it. I'll reteach it to you. Like that's the vibe of so many labs. Like we don't assume that you're coming in with skills. We're planning to teach you. Like that's part of our job as professors. And so, yeah. And just a second, join the lab where everyone's really nice and kind. There's no point in being in toxic spaces. Just shout out that.
Yeah, so I agree with all the points that have been covered by Jason, Kevin, and Jess. And thank you, Jess, for bringing out that it is very important to join a lab where people are nice and kind and are willing to share their knowledge with others, because that's how we build up a nice research environment where students can feel confident that they can express themselves. They can ask questions that they may think that it's stupid, but they are really not, because we, having done so many years of research, don't really understand that when we started, we didn't know how to streak colonies that would space out individually, or whether we are planting out plants, how do we label them properly that they don't get mixed up? When do we have to harvest seeds from a single, or like if someone is maintaining flies, then they don't have to mix up things or whatever. So I think it's very important that knowing that you will be working with colleagues that are nice and willing to share. And then one thing that I would like to add from my side is, no matter what, never hesitate to ask questions when you're working, no matter if you think it's really silly, but no, you will regret later in your life not asking, but you will regret very less asking it straight away and then learning from them.
Thank you all. That was a great discussion. We'll move on to the next question here. And in fact, a lot of questions have been kind of chattering around this idea of hands-on research and just hands-on experience in general. And say a student is able to find a lab that they're interested in an academic setting, or even if they hadn't yet, there's some questions around in an academic environment, if there's enough exposure for undergraduates to get the amount of hands-on research that will help benefit them down the road. What would you all say is the benefit in looking outside of academic realms, say in industry internships? There's a lot of questions around internships in the chat in general. What are your perspectives there regarding looking outside of academic labs? And Dr. Cox, we can start with you.
Yeah, I say like it's kind of like my first point. It's like keep your options open. I think like looking at academic settings, it might be a little bit easier just because you're already undergrad that's like within this body of environment, so it's easier to kind of go and talk to professors that you already interact with. But that shouldn't stop you to say like if you want to explore industry, and I'm not the best guy, maybe Jason can provide more of a point into this, but like there's definitely opportunities for internships for like to just go to an industry for a summer or a company for a summer and just kind of see like what they do. So yeah, I think that's definitely a viable option.
Yeah, there are a lot of opportunities out there. Hopefully, when the world calms down, the government internships or those experiences are going to be available as well, right, outside of academia. But in terms of industry, lots of great opportunities. Summer 3-month internships, you get paid, you get great research experience, and so I want to get let you know that you got to plan ahead for these things, right? So the window for applying for internship programs is generally starts in September, October, in that area for the following summer. So you cannot start planning now for the coming summer for these kinds of internship experiences. But if you do think ahead, say, hey, okay, this is first semester of my junior year. I want to get an experience between junior and senior year. Start thinking about that. So start planning, start looking at career pages at whatever organizations that you're interested in, right? Might be Corteva, you go to the careers page. Around September, you start searching for, they filter out in the careers page for internships. You start looking, see what's available. And don't count yourself out because you think it doesn't perfectly describe you, right? Apply to things and see what opportunities might come up.
Yeah, I don't think I have a ton to add. I think I agree with Kevin and Jason, so I'll just skip this one.
Yeah, likewise. I think, yeah, all I know is from experience from my colleagues whom I know that they did do a little bit of internship in industry, and they did get an idea of how things work in industry. And then they also got a chance to work as interns in research labs, be it as their even a few weeks or something. And this helps them to get an idea of what their focus or what their research interests align really well, and then they move forward with that. But that doesn't mean that it blocks the path for the others. So there are always ways to return what like Jason showed very beautifully. So I've seen many people going to industry, then coming back to academia, and then vice versa. So yeah, it's any experience always is useful.
Thank you all. And the next question that I'm seeing a lot about is the big unknown of funding and the big scary part of and world of funding. And we have several questions in the chat or just asking about suggestions for how to approach this, because obviously this is a very uncertain and frightening realm of getting involved in research. Jess, if you wouldn't mind going first with this, if you have anything to say on this, and I'd love for you to start.
Sure. Yes, this is a very hard time, especially in the US. And this is what I tell my students is that we need scientists and people who can communicate about science now more than ever. And so it's definitely, I think, a harder landscape to get positions like than it was when I was applying because of the state of national funding and investments. But that said, like, I really want to encourage you not to give up, I think, in that way. Like, I feel really strongly that we need more scientists now that more than ever. And in terms of like actual positions, I know, like, I was just looking, I mentioned it in my presentation, but like Neon is this great organization where they hire a bunch of summer employees to go all over the country to work at ecological field stations, and they're still very much hiring. So there's definitely a lot of places that are still working and moving forward. And a lot of the summer programs at institutions, like I know at my institution, a lot of it is coming from private sources that aren't federal. And this kind of goes to Jason's beautiful figure where he was showing how it's all very much interconnected and there are different sources of funding that are beyond federal in the US. So that's what I'll say.
And if any of the other speakers, if you have anything else you'd like to add or jump in with, feel free.
So I think my additional point to that was you need to, more than ever, you need to be very open to things that you maybe wouldn't have otherwise considered. You need to be exploring a wide space, search area for opportunities. If you think, okay, this is the only way I'm going to go, I'm only going to go in this direction, I'm only going to apply for these funds, that's a very risky thing to do these days. You need to be searching all the opportunities out there and finding things that maybe you didn't think were exactly what you were thinking about, but maybe it gives you a bridge over into the next stage of what you're going to do. So.
Awesome. And we have just a few minutes left here. Oh, Dr. Bhatia, I don't know if you were going to add it.
So I only wanted to add one thing to it, that I do acknowledge that funding situation is not the same in every country and also every institution. Some institutions are very rich and they do have internal funding to support these kind of internships. Some are not, but this doesn't mean that people should hold back in reaching out to scientists personally and not waiting on for specific open calls. Because I see that at least at Sainsbury, I receive many emails about, and people also receive emails about if there are possibilities of doing some sort of, like, even for a few weeks to learn some experiments. And then this helps them give an idea of whether they want to do this for long term or not, just to get a feel of how is it to be in a research lab. So start with something very simple in case where opportunities are limited. Reach out to scientists if they can accommodate you in the lab, even for a few days or week, where you do get an exposure that will also be very valuable in the future.
Awesome. Thank you all so much. Looks like we just have 1 minute left here, but I guess I'll just leave the floor open. If any of our speakers, if you have any last minute thoughts or quick second takeaways for our audience, I'll leave the floor open.
I was just going to say, I'm about to put my at least my email address in the chat. These are very short conversations, but I'm sure I speak for the panel that reach out to us. It's so easy to set up a video call with us. Say, hey, I just like 15 minutes, half an hour of your time just to talk about what you do or what opportunities you have, or just get advice. reach out to us, set up those opportunities. We're around. We're busy people, but we'll always make time for students.
Well spoken. Thank you, Dr. Rauscher. And I appreciate all your speakers, all of your advice and responses to all these questions. We've had a lot that have come through the chat. So thank you for your incredible presentations today.
Thank you, everyone, for joining. Again, a big shout out to all our speakers for sharing your insight and sharing your story, and also kind of helping inspire many of our audience today. Hopefully, to our audience, you found kind of listening to the stories and experiences of our speakers today kind of help you kind of at least figure out what to do next. Is always a big challenge, right? You don't have to have everything figured out, but hopefully this kind of help you get there and help you with that process. And with that, again, a big shout out to our Plantae Fellows who organized today's webinar. And I hope that everyone involved kind of find this time valuable. And to our audience, we hope to see you on the next one. And I hope that you all have a wonderful rest of the week and a great weekend. Have a great day, everyone. Bye, everyone.